So we're going to be looking at, so these aren't all of the fungi. I've selected a few of them. So on Practical 4, you should have this list of all the organisms. And if you look here, at the end of that list are all the things you need to know for the different organisms, right? I also have a matrix for you all that have all the organisms on it, and it's available for you already in underneath that Practical 4 unit, okay? So you can get it if you just simply will pick it up. It says, you know, unit 4, and that's what we're going to talk about today, okay? All right, so remember, when we talk about the fungi, it's really about the mycelium and the asexual spore formation. That's how you identify them. And then there might be some specialized traits that really are diagnostic for the organism. And I'm going to show you those so you'll be able to identify these organisms pretty quickly, right? So remember the entire lab was set up based on the 3 M's: morphology, which was unit 1, right? Metabolism, which was unit 2 and somewhat unit 3, but then molecular techniques, diagnostics was unit 3, practical 3. And now we're back to morphology. We're back to looking at the eukaryotic organisms, right? There are numerous books that are published on the description of the medically important fungi. And so I am a trained mycologist. In clinical practice, this is what I did. The organisms you're going to be responsible for, there's 2 yeast, right? Candida albicans and Saccharomyces cerevisiae. There is 1 Zygomakota, Rhizopus solonifer. And then there are 4 different species of Ascomycota. And those are the ones that we really worry about: the Aspergillusniger, Penicillium notatum, Microsporum canis, causative agent of the ringworm, one of them, and Coccidioides immitis. I didn't put all of the true pathogens in there. I didn't see it necessary. I wanted you to kind of have an overview of the entire fungal kingdom that are medically relevant. And so I just didn't do every single organism just to be mean, right? I just selected a few. Okay. So let's look at these organisms. And first of all, Saccharomyces cerevisiae, there isn't anything special about it, right? Except that it can make really great beer, right? Cerveza. So this is a yeast that's used in brewing and a bunch of other things. But the way you can tell yeast are that they bud. And so if you see budding, it's going to be yeast. And so that might be a question, right? You might look at this and I might say, what organism is this in general? And then you would select, because I'm going to make a multiple choice because you like that. You're going to select yeast because it's budding, right? And so here you can see my red arrows are pointing to two different microscope slides. One of them is showing an unstained yeast, and the other one is showing stained yeast, right? And so you can see that the budding process is very, very easy to see. Does anybody have any questions about how you identify yeast? Real easy. Okay. All right. Candida albicans is a little bit more complicated, but it's easy also, right? So you're going to see budding yeast at the kind of yellow arrow, and then you're going to see pseudohyphae. The pseudohyphae are the elongated structures. Those are yeast that are elongated, right? So if you see budding yeast and pseudohyphae, then it is Candida albicans, right? Where Saccharomyces cerevisiae has no real clinical relevance, it's just a yeast. Saccharomyces cerevisiae has no clinical relevance. Candida albicans has clinical relevance. So you can see the 2 top images are showing oral thrush, right? And then the bottom one is showing vaginitis, right? Showing an overgrowth of the yeast in the vaginal cavity, and then you can see the cervix there also. All right. Any questions? All right. Let's move on to talk about the Zygomakota. So the Zygomakota, I've selected Rhizopus solonifer mostly because it's easy to identify, right? It is a less evolved organism. It has aseptate mycelia, But the giveaway is at the red arrow, and those things that look like roots are called rhizoids, and they are used by the fungus to attach to different substrate and different strata, right? So that's easy. That's an easy diagnostic thing to see, right? And so sometimes if we ever get to work together, I will a lot of times just simply do this, and that's diagnostic, and that means that that is diagnostic for that particular organism or those rhizoids. So Rhizopus stolonifer can cause skin infections, eye infections, ear infections, and lung infections, right? But for the most part, it doesn't really cause us problems, because if we have a competent immune system, it's not a problem typically. Okay? Any questions? All right, move on to the Ascomycota. And we look at Penicillium notatum. Penicillium notatum looks to me at the pink arrows like it's a hand, like the phalanges of the hand, or a brush or a broom, right? And then off of that are the Coccidiospores, right? So that's an easy way to remember. It looks like a brush or a broom or a hand. I might put one of those in the description. I'm going to say, identify this organism. It resembles a hand or a brush or a broom, right? And that's Penicillium notatum. So the organism is more evolved. It's got septate mycelia, right? And so you can see the septations here. You can see the septations here, right? They're different cross walls, right? You can see the septa right there also. The mycelia gives rise to these things, these structures that are called phyalides. The phyalides are kind of, if you will, the hand. They're the phalanges of the hand. And then the phyalides give rise to the Coccidiospores. Okay? Any questions? The next organism gets confused with penicillin, but they're not alike at all, right? So this is Aspergillusniger, and it has a vesicle, but the vesicle gives rise to the phyalides, and then the Coccidiospores are produced off of the phyalides, right? But the most important thing that really distinguishes, besides it has a vesicle, right? The most important thing that gives it away is it has a foot cell at the bottom or the base of the mycelia. So here you can see the foot cell, right? Here, this is a drawing by this young lady, Aline Rojas, right? Don't know her, but I liked her drawing, so I used it. And then here's a foot cell of a microscopic image of the organism, right? So if you see a foot cell, and I will show you a foot cell, right? So you want to look at the base of that fungus, right? Because it's either be a foot cell or it will be rhizoids, and that'll be really helpful for us to be able to tell them. Now, these organisms, Penicillium and Aspergillus, can cause infection of the skin, the lungs, the ears, and the eyes, right? But for most people, they don't, because we have a competent immune system. So real quickly, the way you can tell them apart is you can see that Penicillium has these mycelia that gives rise to phyalides, and then the phyalides give rise to the Coccidiospores. The mycelia on Aspergillus gives rise to a vesicle, and the vesicle gives rise to the phyalides, which then gives rise to the Coccidiospores. Okay? And that's how you tell them apart. Any questions? Okay. One of the easiest organisms to identify, hence I selected it, is Microsporum canis. It's one of the etiologies that causes ringworm. And so it looks like a leaf. It has one terminal conidia, and it looks like a leaf. And so it's real easy to identify. So if you see this organism looks like a leaf, it's going to be Microsporum. Okay. And then you need to know that it causes ringworm or tinea, right? And so here you can see this is tinea corpus, right, of the babies. I don't know how that baby got it, but he's got ringworm all over him, probably a cat. And then here you can see this is tinea capitis. It's got ringworm of a scalp. Okay. Questions? All right. All right. Last one for the fungi is Coccidioides immitis. I selected it because it's the bad boy of the entire group. Now, it has a lot of different stages. I selected the oidic stage, oidia. Oidia is defined as the thickening of the hyphal elements within the mycelia, and then these particular hyphal elements become spores. They become arthroconidia, and so they get disseminated, and therefore people can breathe them in, or they can get into the skin and cause all kinds of problems. So you should be able to, on the practical, identify oidia, know that that's associated with Coccidioides immitis. There are other parts of the life cycle. I'm just showing you this one, right? I'm not going to show you others, just this one, because it makes it easy to identify. Okay. And then, it can cause infections of the lungs and the skin. Okay. Any questions about the fungi? Spell what, James? Oh,
Sorry, you said oidia and something else that's
uh-huh.
not on the slide, so I don't know how to spell that stuff.
Okay.
Oh wait, it is on the slide. I'm sorry. Yes.
Okay. Anybody else? All right. It's also, hey James, it's also on the matrix. All right. Protozoal, right? Protozoal. So we're going to be looking at the protozoal parasites today. And so remember, it's all about the fecal oral route. It's about the cyst and the trophozoite, and then the infections it causes, right? And so if we take a look at them, we're going to be looking at the archaeozoa, the euglenozoa, the amoebozoa, the Pseudophora, and the apicomplexa, right? On the organisms that you'll be responsible for the archaeozoa, Trichomonas vaginalis and Giardia lamblia. For the euglenozoa, Trypanosoma brucei gambiense and Trypanosoma brucei. For the amoebozoa, Amoeba Proteus and Entamoeba Histolytica. For the Pseudophora, Paramecium caudatum and Valinium choli. And for the apicomplexa, Plasmodium falciparum, Toxoplasma gondii, and Cryptosporidium parvum. Okay, that's it. So let's go. So the archaeozoa. So this is Trichomonas vaginalis. To me, it looks like a mouse click click, like the mouse that's associated with your computer, right? It's oval in shape. It has an axil style, right? This is the axil style right here, and that's the way it attaches to the surfaces of the tissues it's infecting. So you can see here, this is a cervix. We call this strawberry cervix. It's an indication that this particular person, this lady, has trichomoniasis, right? So it causes an STI, sexually transmitted infection, right? Vaginitis, balanitis in men, but it's consistent with strawberry cervix, right? It only has a trophozoitic state because it goes from the penis to the vagina, and there are no hostilities. It's not a hostile environment like the GI tract would be. So it doesn't really need a cyst state. It only has a trophozoitic state. Okay? Questions? All right. Giardia. So Giardia, we've talked a little bit about this already, so it should be kind of a, hey, you remember this? So Giardia has a cyst and a troph. You're responsible for both of them, right? Belongs to the archaeozoa. The cyst is round. I'm sorry. The cyst is oval in shape and has 4 nuclei in them, right? You can count those 4 nuclei. There's 1, 2, 3, 4, right? And those nuclei, when the cyst breaks apart, they become trophozoites, right? The trophozoites to me resemble a kite or a flounder, right? Or a skate, a fish, a skate. And those things that look like eyes, remember, are not eyes, they're sucking disc, right? And so they're easy to recognize. And this organism causes a form of dysentery. It's a mild form of dysentery, but it's still dysentery. Okay? Questions? Oval in shape is a cyst. The trophozoite looks like a kite. Okay? All right. Move on to the Euglenozoa. So the Euglenozoa, are the hemoflagellates. And so you can't really tell them apart by looking at them in a blood smear. What you can tell, it's a hemoflagellate, right? So if you're looking at this particular image, look at the purple arrow, you can see the trypanosome in blood, right? It's out there outside of the blood cells, and they're about the size of 2 to 3 erythrocytes. And I might give you that clue. But the way that you're going to be able to differentiate them is I'm going to tell you the part of the world that they were recovered from, right? So if I say that they were recovered from South America or Mexico or the United States, it's going to be Trypanosoma cruzi, which causes Chagas disease, right? And the vector is the rejuveate bug, the kissing bug, okay? If I say it's from the African continent, right? Then of course it's going to be Trypanosoma brucei or Trypanosoma gambiense, and the vector, of course, is a tsetse fly. Okay, that's all you have to know for these guys. All right, Entamoeba histolytica. It's round and it has at least 4 nuclei, right? So this is different than Giardia. Giardia is oval in shape. This is round, right? And I will probably give you that clue. I'll say this is a round structure, right? The other thing I want you to know is that if you see the matrix, and the matrix has all this debris in it, right? So you see all this debris, then it's coming from a fecal smear. The other matrix you're going to see is blood, right? So it'll give you some hints that way so that you'll be able to tell what organism it might be, okay? The other hint that you might see from the trophozoite is that the amoeba have no real shape. So if you see the word amorphous associated with anything, you know that if it's amorphous, it has no real shape, right? There's the word amorphous. And if you see that word from me, then you know you're going to be dealing with an amoeba, okay? Entamoeba histolytica causes dysentery, amoebic dysentery. Okay, any questions? All right. Amoeba Proteus is a commensal, and I'm mostly going to use it to talk about pseudopoda, right? So if I have an arrow saying what are these things, then you're going to say it's a pseudopod or pseudopoda, right? But Amoeba Proteus, I will show it to you like this. It'll have all these pseudopoda, right? And so you're only going to need to know the trophozoite for this organism, okay? Another commensal, Celophora, is Paramecium. And so I'm mostly going to show this because you can see the cilia, right? But this is Paramecium. It doesn't really cause a problem. So I will only ask questions about this, and I might ask you what's a genus or the class. Celophora, it's Paramecium. It's a genus. And then I might point to the cilia and ask you what these things are, and you'll say cilia. Okay. There is one infectious agent in the Celophora that is important, and that is Valinium choli. And it has both the cyst, which is perfectly round, and the troph, which is more oval in shape, have a macronucleus. And the macronucleus is always, to me, looks like a U or a horseshoe. Okay. So even though you can't see the entire macronucleus here, because this is a 3-dimensional organism and we're looking at a one plane, it's there just like that. Looks like a U or a horseshoe, right? And the same thing here, it's there. And here it's easy to see, okay? So this also causes dysentery. The cyst is perfectly round, has a macronucleus. If you hear the word macronucleus or you see the word macronucleus in the text for the question, it's going to be Valinium. Okay, perfectly round, the cyst, oval in shape, the troph. And if you look real close, you can see the fuzziness associated with the troph, and those are the cilia. Okay, last group, the apicomplexa. And so Plasmodium, I think, is going to be the easiest one of them all because it's the only one found inside of the red blood cell, and I will show it to you inside a red blood cell. And it's going to look like a ring, right? So if you think about this, then here, if you think about the way I draw this, right, there's the ring, and there's the big old diamond here, right? And so that's what they look like. So we call them a ring structure. They're also the trophozoite or the merozoite. They all mean the same thing, right? So all those terms mean the same thing, right? Ring structure, trophozoite, merozoite, they all mean the same thing. Okay. And it's inside a red blood cell. It causes Plasmodium causes malaria. Okay. Any questions? Okie doke. Toxoplasma gondii is the most complicated of the organisms we'll see today. It has two different stages of development. It has a trophozoite and it has a cyst. Inside the cyst, which is perfectly round, are a bunch of structures called the bradyzoites. You need to know that. All these little things inside of there are the bradyzoites. The trophozoites, if you look at them, they're known as the tachyzoites. And remember, they look like a canoe or a banana with a single nucleus, right? So these can cause all kinds of infections, right? They get a lot of discussion because of pregnant females and causing abortion in them, but really, it can cause all kinds of septic infections, parasitemia, all kinds of different things. And the known reservoirs are cats and birds, right? You should know that also. Okay. And then the last thing for today is Cryptosporidium parvum. This one's going to be easy because it is the only organism that I've selected in the protozoal parasites that is acid fast. And I will only show you the acid fast organism. So hot pink, this is when you can remember hot pink, is going to be Cryptosporidium parvum. Now here are the cyst and the trophozoites here, but those are really hard to tell because they don't. I mean, morphologically they look like a lot of other things. So I just thought it'd be easier for everybody if I just showed you the acid fast organisms. Look at the matrix. You can see that the matrix has all this debris. Mercy, what does all that debris tell you? Anybody can answer. That it's from feces, right? Right. So not an amoeba, but that it's associated with feces, all that debris. So therefore, it's going to cause a form of diarrhea. Now, people say it causes dysentery, but it's not as bad as dysentery. Although I've never had it, so I guess I can't really say that. But it's going to be diarrhea or dysentery. Okay. Any questions? Cryptosporidium parvum causes dysentery. Okay. Acid fast. A little bit of artwork. Any questions about anything we did today in lab? Okay. So